Welcome everybody. It's really great to see you here. Thank you all for tuning in for this BME symposium and the beginning of it, where our first session is on imaging. I'm just here to sort of provide some initial thank yous across the board. The BME department has roughly 30 core faculty and roughly 100 affiliate faculty sort of across campus in engineering and medicine and various other schools and colleges across the country. And so we thought that having an annual event in which we try to sort of bring together this really diverse group of people would have a very good purpose, and particularly given the pandemic and everything that's happened to sort of disconnect our community, we thought it even more important to have an event where we could actually gather around something that we're really passionate about, which is science. So I really wanted to sort of thank, in this case, Doug and Zhongming for really organizing this session that we're about to have, and as well as all the other organizers that are throughout the next couple of days. The other thing, in addition to having the symposium, is I really wanted to use this as an opportunity to sort of recognize some of the things that have happened within BME over the course of the past couple of years as well too, and sort of use this as an opportunity to update people on some things that are going on. And one of those you might have heard is I sent out a request for nominations for 2 awards, and so I'll just jump right to these awards and sort of want to recognize the 2 awards, one for the DEI and one for mentorship. And so congratulations to Chris Tossas for the DEI award and to Brendan Baker for the mentorship award. You can see in the statements here in terms of that I've selected from the nominations that sort of recognize why people have been nominated for these awards. It was really phenomenal to see the number of nominations that came in, and really how exciting our community is in terms of trying to recognize exciting developments. Chris in particular has been a member of the DEI committee and has really worked tirelessly for the past several months to really promote and oversee these programs to essentially bring up the equity issues and having discussions around them. And then also congratulations to Brendan Baker, who is really an assistant professor in the department. But you can see here the students that he's worked with have been really excited about what he's done to sort of provide training to essentially help them launch their careers. So really a testament to the effort that both Brendan as well as the students have put forth. So congratulations both to Chris and to Brendan. And want to say thank you all for what you do, and also to all the nominators in general. I want to say thank you for putting forth the names and really recognizing what a great community we have in general. Sort of moving on, I thought I would mention as well just other things that are in the news. We can see that Xiaotian Wang has been recognized for his works in physics world, highlighting the work that he's done with real-time dosimetry. We've got other work by Sherman Fan, who has responded very with great effort, trying to work on better treatments for COVID, or I would say for detection of COVID and monitoring lung injury along the way. Congratulations, Sherman. It's really been an extremely exciting project to sort of being able to follow over time. Zhen Xu, who has received the Lockhart Prize, it has been a couple of years now, but we've sort of missed the opportunity to celebrate this when we didn't have this symposium last year. And similarly, we didn't get to celebrate this last year when Xiaotian Wang was inducted into the AIMBE College of Fellows last year as well too. And so it's while we couldn't do this in 2020, I'm glad that we could actually do something virtually this year to really sort of acknowledge some of the great events that are going on. So with that, I will turn the floor over to Doug Knoll, because you all wanted to come in to hear more science and imaging. And so Doug, please take it away.
Great, Lonnie. So congrats to our DEI and mentorship awardees. This is the first year I think that we've done this, and I'm very excited for the awardees. Also, congrats to the other awardees. We're going to try to get rolling pretty quickly. The schedule is tight, and so this is an opportunity to kind of showcase some of the things that are going on at Michigan in respect to imaging. This is historically has been one of Michigan's strengths. We've been long-standing collaborations between radiology and biomedical engineering, and long-standing, long list of researchers over the years in PET, in ultrasound, and MRI, and more recently in things like photoacoustics and all. And so I'm really looking forward to hearing the talks today. Our first speaker is Professor Zhongming Liu, and he is a relatively new faculty here at Michigan. Many of you have not met him yet, in part because of COVID. He's actually been here about a year and 4 months, but unfortunately, we've been locked down for about a year and 2 months of that period. So he got his PhD, University of Minnesota, under the direction of BME, but also worked closely with the MR facility there. Did a postdoc in functional MRI at NIH, and was at Purdue for about 6 or 7 years, and then he joined us here, like I said, about a year and 4 months ago. So I'm really looking to your talk, Zhongming, please.
Thank you, Doug. Let me share my slides. I hope that slides is displayed properly.
Perfect.
Okay. Thanks for the introduction. Really thankful for this opportunity to share my research. Indeed, very regretful that I was not able to interact with many of you because of the COVID. So my lab does a different aspect of imaging, and one area of imaging is trying to use multimodal imaging and recording methods to map brain functions and model brain functions. Another area of research is trying to combine data science advances. So we're trying to develop a brain-inspired deep neural networks for two purposes. One is to decode brain activity. The other is trying to make those networks smarter and to do human-like tasks. And today I will talk about the third area of research that we have been really into in the past several years, that is to use MRI to investigate gut-brain interactions. There are basically two topics I would like to touch on. One is to use MRI to image the gut and image the brain and their interactions. The other is to apply neuromodulation to stimulate the gut-brain neural axis. And then finally, I will touch on some future directions that we are heading to. We're interested in the gut and the brain. Gut is more than just is a very special visceral organ. And some people say that the gut contains a second brain. There are even folks who said that the gut is the first brain, and the brain is actually a cognitive silo of the gut. The gut has a very extensive nervous system, like the brain's central nervous system. This nervous system in the gut is called the enteric nervous system. One of my collaborators, John Furness, has been studying for several decades on the enteric nervous system. And the enteric nervous system, central nervous systems are interconnected through the peripheral nerves. And another collaborator of mine has been spending decades of mapping the extrinsic peripheral nerves innervation that innervate the gut from the brain. So we're interested in the gut-brain interaction specifically through the neural axis connecting between the gut and the brain. And there's one pathway for the neural axis is the vagal pathway. The other one is the sympathetic spinal pathway. So most of my research involves only the vagal pathway. So we have three nervous systems: the enteric nervous system that resides in the gut, and the peripheral nervous system that connect the enteric nervous system to the central nervous system. Over the years, we have begun to understand a lot more about all these three components that are involved in the gut-brain interaction. So in my lab, we primarily use MRI. So we have developed an array of technologies, including functional MRI to look at the brain responses, activations, and the connectivity. We have neurophysiology to look at neuronal recordings, neuronal activity in the brainstem, specifically the dorsal vagal complex. We also use MRI to trace for functional and anatomical assessments of the nerve activity passing through the vagus. And we use MRI to look at the motor functions of the stomach and the intestines, and using electrophysiology to look at the gastric pacemaking activity. Now, first, to use MRI to see the stomach, we develop a protocol that we train animal, we fast the animal and train the animal to voluntarily consume a diet gel and label the diet gel with gadolinium. So all the food and the meal and nutrients becomes visible inside the stomach. This is compelling because this provides a way for us to see a somewhat irregular movement, complex movement throughout the entire GI tract non-invasively using MRI. And this technique is translatable to humans. So for example, in humans, we don't use gadolinium, and we can use all natural ingredients to mix a meal, test a meal. And this is made with firm tofu, blueberry, and the pineapple juice and the bananas. And this ingredients is very rich in manganese, which is a T1 shortening contrast like gadolinium. So then we can take a fast MR images of the stomach and the GI tract, so we can start to see the motor functions of the stomach. And then we can apply image processing techniques to look at the moving patterns, the contraction patterns of the stomach, and analyze these contraction patterns using phase synchrony to see how the contraction is coordinated or discoordinated. And this can be done in individual animals or patients. And we know the gut does not just function by itself; it interacts with the brain. In fact, that when the gut has this periodic and wave-like contraction, and the whole phenomena is also monitored by the brain. And to look at that, we record the gastric electrical activity, and then look at the synchronization between this peristaltic activity with the brain signals measured with MRI. We have found that there are a number of brain regions that are actively involved in listening to the gastric rhythms and likely monitor the gastric rhythms. And this is most likely engaged through the vagus, because when we cut the vagus, most of this brain-gut synchronization disappeared. We also try to use MRI to assess the nerve activity. How do we do that? We inject manganese chloride, calcium surrogate that are visible to MR, and then we inject this to the nodose ganglion, which is where the afferent neurons are sitting, and the afferent neurons are activated by intrinsic activity or the nerve stimulation, and then transport the manganese into the brain, where we can assess where they projected to. So this is a non-invasive MR-based neuronal tracing. We find out that when we deliver the vagal nerve stimulation, we can promote this afferent activity and result in more transport of the manganese into the brainstem. So this is very intuitive then. So we actually start to stimulate the vagus nerve. When we stimulate the vagus nerve with a specific parameter that we find to be effective, and it shows that this stimulation can promote gastric emptying specifically by opening the pyloric sphincters longer and wider. Stimulation is very sensitive to the parameters that we choose to use. If we target the afferent stimulation, then we are not going to disrupt the intrinsic pace of the stimulation, but we modulate its amplitude. But when we actually stimulate the efferents, we can find that sometimes when the stimulation is on, it disrupts the pacing, the rhythm, and stall the contraction patterns. So it can be really bad. Then we start to use fMRI to see what would be a good stimulation, what would be bad stimulation. It turns out that the stimulation, when it works, it all engages a lot of different areas in the brain. So engaging the brain is a key to effective stimulation of the GI function. In addition to fMRI, we also use neurophysiology. So we put electrodes into the brainstem and targeting at the NTAs, and we apply stimulation to the stomach wall and trying to engage the vagal afferent nerves. And we have really nice signals recorded for multi-unit activity, and we can do spike sorting to assess single unit activity as well. And surprisingly, what we find that it's not only where you want to apply the stimulation and what amplitude you want to apply the stimulation, the frequency, other parameters like what people have used, and also in the clinical approved devices, but also the direction of the stimulation is also very important. It turns out that if we want to engage the brain effectively, we need to apply the stimulation in a direction along where these receptors are aligned to. So in this case, it's a longitudinal RMA receptor of the vagal afferents. We apply the currents in the same direction of those receptors, then we got a really strong vagal afferent responses recorded from the brainstem. If we apply different directions, we almost see no responses. Well, to conclude, quickly wrap up, and we have developed this MR technique. We believe it's really useful and provide comprehensive assessments of gastric motor functions. And fMRI and EGG recordings would be able to use to report intrinsic brain-gut coupling. And vagal nerve stimulation, gastric electrical stimulation may optimally modulate gastric motility and emptying through selective engagement of vagal afferents. We are really positive about this direction, and we think that combining MRI, fMRI, and electrophysiology can inform where to stimulate and how to stimulate for treating gastric disorders. Furthermore, we'll start beginning to look at a mechanistic question. So we actually launched a new project that we're going to look at the model of the stomach, including its enteric nervous systems and extrinsic neuromodulations. This project is currently ongoing, and it involves multiple institutions. We also have a new project that we launched here at Michigan with Michigan Medicine, specifically Professor Jianzhe Chen and Ouyang Chun and some collaborators from Penn State. And we're actually going to look at all the functional neural circuits are involved in the brain-gut interaction, including the vagal pathway on the peripheral side and the central gastric network in the central side. And we're about to start this project in the coming, maybe July. With that, I would like to thank my teammates and Dr. Schingler and my student Cherry and Shaokai and some of my former students at Purdue. With that, I'll thank for your attention. I'll be happy to answer any questions if there's any, or we can also move on to the next.
Thanks, Zhongming. That was fabulous. First, I give the opportunity if the audience, if someone has any questions, you can either type it in or unmute. This is a Zoom meeting, so we can unmute and ask questions that way if you want. So Zhongming, let me ask you this question, because I know it's one you've thought about a little bit, which is that what are the prospects for translating some of this work to humans? I know you talked about the sort of the naturally contrasting meal that you could have humans eat, but what about the electrical circuitry? Do we have ways of monitoring that? How accessible is the vagus nerve in humans, and is that going to be useful in any kind of neuroimaging studies? What are your thoughts?
Right, and we definitely have more space to run invasive recordings in animals, and in humans we will plan to look at maybe auricular branch vagal nerve stimulation or transcutaneous vagal nerve stimulation, those non-invasive stimulators. And on electrophysiology, we also will go to non-invasive recordings, maybe by recording from the body surface, and the signal quality will not be as ideal. One of the major directions I'm hoping to head to is to develop MR methods to image the gut and the brain in the same time. And that's something that we'll first try out in animals, We're still working on it, but it will be really exciting to use an all MRI methods to look at gut-brain interactions in humans.
There's a question from the audience about are there any specific GI or disorders that you think would be particularly
right? And I'm not a medical doctor, so some of those questions I really defer to my collaborators in Michigan Medicine. And currently, we have interest to look at gastroparesis and functional dyspepsia, and there are also some neurological disorders that accompany those gastric disorders like depression and Parkinson's disease. So there are a lot of brain disorders and gut disorders. They're just of some shared or even common roots. So those are the questions that we have interest looking into.
Fabulous. Great. Okay. So I think, you know, in the interest of staying on schedule, we should move along. It's my pleasure to introduce another relatively new faculty to Michigan, which is Nicole Seiberlich, who's been here about 2 years now. And Nicole got her PhD in Germany with the University of later did a postdoc at Case Western, became a faculty member in biomedical engineering at Case Western, is now here in radiology where she co-directs the MIT, which is the Michigan Institute for Imaging Technology and Translation. Love the name. And with no further ado, I'll let Nicole take it away. Thanks, Nicole.
Great, thank you, Doug. I'm assuming you guys can see my slides and will let me know if you can't. Perfect. As Doug said, I'm an associate professor in the Department of Radiology, and I do have a secondary appointment in biomedical engineering, and I'm really delighted for the invitation to come and speak with you guys today at this BME symposium. I think we have a good relationship between our two departments, and so it's really wonderful to be here. Before I get going, I do want to disclose that my lab receives research support from Siemens Healthineers, and I also receive royalties for magnetic resonance fingerprinting, which I'll be talking about during this presentation. Okay, so we're going to take a little bit of a jump and switch gears to talk about clinical MRI. So what I'm showing you on this slide is an example of a clinical MRI image of the heart. So here you can see the left ventricle, this is the right ventricle, this is the lungs. You'll be seeing a lot of these in this talk. And like most of the images that we collect as part of a clinical exam, this image is qualitative. And what that means is that the brightness, like the brightness of this pixel and the darkness of this pixel, that will help us distinguish different kinds of tissues, but we're not making a measurement of any particular tissue property. We use tissue properties to generate this contrast, but we don't have any units associated with this image. These are just pixel intensities. It is possible to make maps of tissue properties like this T1 map that you see on the right-hand side. T1 is one of the properties that we can measure using MRI. And here you see that the pixel intensities now are related to actual measurements, which you can see by the scale on the right-hand side. And there's a number of reasons that we might want to move to making these quantitative tissue property maps. Really, our goal is that we will scan a whole bunch of people. We'll know that some of them are healthy, some of them are diseased. We'll make measurements of these tissue properties like T1 down here, and we'll be able to draw a clear separating line between healthy subjects and patients with disease. And when we make a new measurement on someone who comes into the hospital, we'll be able to use this measurement to objectively say whether the myocardial tissue is healthy or not based on this measurement. This is where we'd like to be. And we can measure a lot more than just this T1 property using MRI. We make measurements of other properties, including T2, extracellular volume fraction, T2 star fat fraction. And importantly, we understand how these different tissue properties change when a patient comes in with a pathology. So different pathologies will have different changes in our tissue properties. And so if we measure these tissue properties, we might be able to tell objectively what's going on with our patients. So going back to our dream, we'd really love to be in a situation where we can make measurements of these multiple tissue properties and based purely on the measurements, be able to say whether the tissue is healthy or to differentiate different kinds of diseases. So this is where we'd love to be, but it's not quite that simple, unfortunately. We see that depending on how we make our measurements, we can get really different information about our tissue properties. So we're going back to the tissue property of T1 here. We've measured this value in a healthy subject using a standard technique known as MOLY. We can see that the T1 in the myocardium is a little bit over a second. But if we make the measurement using a different technique, or we do our parameter estimation from the data in a different way, we'll get very different values for the T1, even in the same healthy subject. Right, so how quantitative are we really if we're not able to repeatedly measure the same value, even if we change our technique? So what this leads to is that different sites with different scanners, different acquisition techniques, different post-processing methods will end up with different ranges, which they would consider healthy for T1, meaning that the ranges of pathology will also be very different. And we can't just pool all of this data together in order to do large studies to understand how these different parameters will change upon disease. And the reason that we get all these different measurements is that when we're trying to measure one tissue property like T1, we have a lot of confounding influences from other factors, the T2 of the tissue, diffusion, perfusion, the subject's heart rate, exactly the pulse sequence we're doing the measurement, the reconstruction. Again, there's a lot of different confounding factors which lead to this problem. And so now we're in a situation where if we see all of this variation, we aren't quite sure if it's due to how we're making our measurement or whether the patient has disease. So if we come in with a new patient, we can't use this measure objectively to figure out what's going on. The other problem is that if we want to scan values beyond just the T1, we'll have to make this measurement in a single breath hold, allow the patient to relax a little bit, have them hold their breath again, make another measurement of another tissue property, wait again, another and another. And these scans start to become very long when we like to look at multiple areas of the heart or measure multiple tissue properties. So all of a sudden, we're really inefficient and we aren't really sure how valuable these measurements are anyway if they're not telling us objective information. So this brings us to the idea of magnetic resonance fingerprinting. So MRF is a method of encoding tissue properties into our signals and then extracting that information out to make these tissue property maps that you see down here. We use fingerprinting as a T1 and T2 mapping technique, but we could potentially fold in all sorts of other tissue properties and measure them simultaneously. I'm just going to very briefly go through how this acquisition works. So here is a schematic of a pulse sequence in fingerprinting. We collect our data in end diastole during a breath hold using a spiral case-based trajectory, which you see over here. And if we would just make an image with one of these data sets, it looks really terrible. It's highly accelerated, super undersampled. These are lots of aliasing artifacts, which make it impossible to see the image. But if we look at one pixel and all these images over time, we'll see that this pixel will have a unique fingerprint. And that fingerprint depends on the T1 and T2 that's actually in that voxel. But now we have to match this thing to figure out what that T1 and T2 are. And to do that, we take the pulse sequence and we throw that into a big physics simulation, the block equations. And we generate a dictionary of all of the possible signal evolutions that we could have gotten for different combinations of T1 and T2 using this pulse sequence. We'll match then the actual fingerprint we did get in the voxel, and that will tell us that the T1 and T2 in that voxel was 500 milliseconds and 50 milliseconds. And if we do that for all of our voxels, we'll end up with these really nice maps. Okay, this is a really awesome idea, right? So what do we do now that we've had this great idea? In order to start translating, we first have to test everything, starting in water bottles to make sure our measurements are accurate. Once we're happy with that, we might move on to scanning healthy subjects. And finally, we'll look at the application of this technique in patients because ultimately we'd like to translate it for clinical use. Of course, every time we move through this process, we'll see that there's some technical improvements we want to make. We might want to optimize our pulse sequence or how we're doing our reconstruction. We might want to refine how we do this acquisition for different applications. And there are all sorts of new features we might want to add, like additional tissue properties. And every time we make a technical improvement, we have to go back to our testing, scanning in normal subjects, maybe looking at different patient populations. And this whole process just keeps going around and around and around. And this is the phase that we're in right now. Okay, so just as an example, this is one of the first tests that we did with this acquisition. We scanned a series of water bottles here. You can see the T1 map of the water bottles and the T2 map of the water bottles. We know what the T1s and T2s are from conventional measurements, and we make sure that the measurements we're getting from fingerprinting match. So we're doing pretty well in our water bottles. That's a great first start. We also know that depending on the subject's heart rate, we can get different measurements of T1 and T2. And so here we artificially tested with our water bottle what happens if we set different heart rates. And again, we're finding our measurements are very stable. This seems like a pretty simple experiment to do, but one that's really essential before we can move on to the next step. We also believe that it shouldn't matter how we make our measurements. So unlike those other T1 mapping techniques that I showed you, we should be able to get the same T1 value no matter what measurement scheme we use. So here you see each of these little symbols here is a slightly different way of running our fingerprinting experiment. And no matter what way we use, we're pretty good at mapping T1. T2, though, is a whole different story. We get very different measurements if we change our pulse sequence. This is bad. This means we're not accounting for something in our physics simulation. So we went back and said, what are we missing? And we added a couple of corrections in order to ensure that no matter how we were running our sequence, we still got stable values for T1 and T2. And this is something that we wouldn't have noticed if we hadn't gone through to do this special double check of our pulse sequence parameters. Okay, so this all looks great. Now it's time to move on to healthy subjects. This is just an example of the fingerprinting T1 map we got in our healthy subject and the corresponding conventional T1 map. So we're doing pretty good. And then of course, while we get this T1 map with fingerprinting, we're also getting the same, or a T2 map at the same time, which also compares really nicely to our conventional T2 map. Now we have one healthy subject. This is great. And now we begin our giant data collection phase. We collect data and more data, maps and maps and maps. Overall, we collected, oops, there's something missing here. We collected 58 subjects worth of data, healthy subjects. And then we start looking at the numbers. First, we're comparing our fingerprinting numbers. These are shown in red for T1 and T2 to the conventional values that we're getting at 3 different positions in the heart. We see that we do have some statistically significant differences compared to our conventional techniques, which we're not surprised about because we know that we get a lot of errors in our conventional techniques. But one thing that we do notice is that the standard deviation of our fingerprinting measurements is higher. We're keeping an eye on this. This is something we would really like to change, but it will require further technical developments that we wouldn't have seen if we hadn't measured so many healthy subjects. We, of course, look at all of these numbers in a million different ways, look at AHA segments. We've done test retests, which shows fingerprinting compares very favorably, but all of these tests need to be done before we can move on to the next phase. And the most important test of all for us is looking at, is asking our radiologists and cardiologists what they think about these maps, if they would ever use them in a clinical setting. So what we did here is we identified a couple of features that we wanted our radiologists and cardiologists to look at and rate our maps, both with fingerprinting and conventional methods on a 5-point Likert scale. Luckily for us, in this case, we were happy to see that fingerprinting was preferred, and that was statistically significant in all cases, except for this one that I highlighted here in yellow. This is the conspicuity of the right ventricular wall in T2 maps. Our resolution is just too bad really to capture that in any method. So we weren't really surprised by that. But without going through this radiologist rating study, we can't be sure that radiologists will be okay with these images. We also did a forced choice comparison study where we said, which map do you like, the one on the right and the left, and counted how many times they selected the fingerprinting map versus the conventional map. Here, fingerprinting was preferred overall in all of these features. But again, the study, it seems simple, but it's really important for that clinical translation piece. Okay, so in addition to scanning at our site, we also need to scan at other sites because that's where we have the problem with quantitative mapping, that we're not getting similar values in other places. So we asked our friends in places like Glasgow, Montreal, and Toronto to also collect maps. And we pooled the numbers, again, looking at the T1s and T2s. We see that no matter where we are, our T1s look pretty stable in healthy subjects. Our T2s are a little jumpy. And again, this is something that we're keeping an eye on and working on technical developments in order to firm up those T2 values. Okay, so now are we ready for clinical adoption? We've scanned healthy subjects like crazy. Well, we go to our physicians and they say, well, are the normal values the same for each person? And we say, well, we're working on it. Did they change over time? And we say, well, I guess we're working on that now too. They ask about patients. Are the normal values different from values you see in pathology? We have to see. Is fingerprinting more efficient? Can I deploy fingerprinting and immediately get a map on the scanner, or do I have to get the data on a memory stick, take it to my computer, do 2 days of processing, and then will it come back? They want to see it immediately. Will my radiologist ever use this because they don't really see the value in it? Does this improve the workflow? What can I do with fingerprinting that I can't do with standard approaches, right? All of these questions, for me as a physicist, I think to myself, I'll just go back to my MATLAB and my coding. But these are all really important questions that we need our clinical collaborators to ask us so that we can move through that translational step. We have started collecting data in patients. These are really anecdotal at this point, but here's an example of a patient with hypertrophic cardiomyopathy. The fingerprinting maps are shown at the top. And of course, we're also still collecting our conventional maps shown at the bottom because we need these for our comparison. We are doing this acquisition of hypertrophic cardiomyopathy not only here in Michigan, but also in Cleveland and also in Toronto with Bent Fintusberger, where we do the same acquisition, looking at the fingerprinting maps and comparing those to the conventional maps in order to see if we're actually getting different values here in this patient with hypertrophic cardiomyopathy than we would in healthy subjects. We have some, again, other patient studies where we're still in the process of data collection, and it seems very anecdotal at this point. These are fingerprinting maps in a healthy subject versus an amyloid patient. And in this case, we see elevated T1 and T2 in the amyloid patient. This is what we expect to see, and it's showing the added value of measuring not just T1, but T2 as well. But this is still a major work in progress. So is MRF ready for clinical use in 2021? Well, where are we now? Let's look at it really starkly. We've scanned 0 patients for clinical purposes. We put this method out, cardiac fingerprinting in 2017, and fingerprinting for the brain was 2013. So this method is 4 or 8 years old, depending on how you look at it. And in the heart, we haven't scanned anyone clinically. We've scanned about 50 patients for pilot testing. This number is getting bigger every day, but imagine all of the different indications we have. We need to scan a lot more patients. And I'd say we scanned about 250 healthy subjects, but a lot of these have been for technical developments. So we can't even pull this data together to tell us really if we're getting stable values from our acquisition. So is it ready for clinical use? Not yet, probably not for a little while still. And I think that is something that as a biomedical engineer is very frustrating, but it's really worthwhile to know that we're making progress and moving in the right direction, especially with our clinical collaborators. So the story will continue. We really believe that quantitative MRI, especially in the form of MRF, is going to be a really great tool. It's used clinically now. It's just a matter of collecting that data to get this into clinical translation. But there's a lot of work that has to happen before this really good idea can move into clinical use. And most of this work requires clinical input. This is not just me with my code and my scanner. This is really radiologists and cardiologists who help us with these images. And so my take-home message is that it's important to talk to your end users when you're working on a translational project like this. You should try to find ways to quantify the value of your new approach. Just because it's new doesn't necessarily mean it's valuable. And test your methods in real life situations. Just because it's working, in your computer or on healthy subjects or on water bottles doesn't mean that it actually will be beneficial for patients. And don't be afraid to take a step back. If you notice that something isn't really perfect on the way to a translation, it's better to address that at the outset before you're pushing a method all the way through to potential clinical use, and then you have to haul it back. So that wasn't maybe the most optimistic talk, but I hope you can look at this talk and say, having a great idea is really one important step towards moving towards a new clinical solution. But there's a lot in between, and this is why we need collaboration between our 2 departments, which is what we're very happy to do. So with that, I just want to thank the team, especially at MINT and everyone who's been collecting data. And thank you guys and BME for the invitation to come and talk today.
Thanks so much, Nicole. That was very cool. It's always glad to see someone who's taking it for the, in a sort of translating business. There's this valley of death. It looks like you're trying to tackle the valley of death in MRI pulse sequences. We have time for maybe one question. If anyone has a quick question, feel free to unmute or type it in. Is that Parag raising his hand?
Yeah, thank you very much for that talk. So I'm in the brain area rather than the heart area. And my question was, how much of this complexity is due to the fact that you have to do cardiac gating and stop the heart from moving? And how much of it is due to T1 and T2 really being an approximation for a multi-exponential relaxation in any given tissue?
Those are great questions. And I think they're 2 completely separate problems, both of which have to be tackled. So the first problem that you're talking about is the fact that the heart is always moving. And the lungs kind of are moving as well. You can stop them for a little while, but not forever. So the fact that we have to contend with respiratory and cardiac motion makes the cardiac fingerprinting acquisition a lot trickier. We don't have the luxury of spending as much time as we want to encode the signals. And so we're really dealing with limited information. So we often use, instead of the pattern matching, which I presented here, low rank reconstructions or other kind of more advanced signal processing techniques to extract that information. But the translational problem and the problem that you're talking about with the multi-exponential or multi-compartment T1s and T2s, that happens everywhere, whether it's in the heart, in the liver, in the brain, and you're kind of alluding to this. So if you're going to make a measurement using other tissue property tech or other tissue mapping techniques, like say just regular look-locker T1 or even a spin echo T1, you're going to end up with slightly different measurements because you're going to be capturing that multi-exponential information in different ways. It's very difficult to separate out different exponentials from each other when you're just dealing with an exponential. That may actually be easier in a technique like fingerprinting. And there's been some work, especially in the brain, looking at multiple compartments, because if you have non-exchanging compartments, you can essentially treat those fingerprints as basically added to one another. They're just a weighted sum of the different compartments. And so fingerprinting does provide an additional opportunity to help us separate those different components out. It's much easier to think about in the brain, again, because you aren't contending with all of this motion. But that's one of the problems that we'd actually love to be able to get to the bottom of, to do a more complex modeling, looking at multiple compartments and exchange between compartments in order to access those different parameters that are really challenging to do with regular mapping techniques and fingerprinting as well.
Thank you. That was that was a rapid fire response. Thank you so much. I don't think it would take us to give that response. Okay, thanks so much. I think we're going to try to move on to the best interest of time. So we're going to switch gears for the next presentation. We have Yannis Paulus from the Department of Ophthalmology. Yannis got his medical degree from Stanford, did an internship at Sloan Kettering, went on to do residency and was chief resident at Stanford, and then and after that came to Michigan, where he's now in the Department of Ophthalmology. So thank you.
Hi, everyone, and welcome. Thank you very much, Doug, and everyone for giving me the opportunity. I'm a retina surgeon and clinician scientist, primarily in the Department of Ophthalmology and also with the dry appointment and BME. And first, I'd want to start with thank you all very much for giving me this opportunity. I'd like to thank Shiding Wang. We've been collaborating together now for 6 years since I've been here, and it's been a very productive collaboration on what we'll be discussing today: multimodal photoacoustic microscopy, OCT, and fluorescence, molecular and cellular imaging of the retina. For disclosures, I do serve as an inventor on 8 University of Michigan patents, a number of which are related to what we'll be discussing today. We've also co-founded some eye imaging companies also related to what we'll be discussing, and these are preclinical systems not approved for human use by the U.S. Food and Drug Administration. So my big interest is on molecular imaging. Anatomic changes are really the end result of complex molecular pathways. And right now, most of our imaging is end-stage anatomic changes. So I'm interested in moving the needle upstream, looking at changes in growth factors, integrins, proteases, so we can have a better sense of the molecular changes taking place in individual patients so that we can practice more precision-focused medicine for each individualized patient in each disease. We're really a world's expert on photoacoustic imaging, so that probably needs no introduction with you all. But some advantages of that, specifically in the eye space, are the deep penetration you get, where we can actually see some of the deeper vasculature called the choroid in the eye, the high resolution, so you get optical strength resolution, non-invasive, high contrast, getting you structural, functional information, and then with contrast agents, molecular information. The primary endogenous and exogenous absorbers on the eye, endogenously hemoglobin, both oxygenated and deoxygenated, and melanin are the 2 main ones on the eye. And for exogenous, the sky's the limit, but I'll be discussing primarily gold nanoparticles since that's what we've been using. Here's a schematic of our setup that has integrated photoacoustic microscopy, optical coherence tomography, fluorescence microscopy, all integrated into one system. And here's how it actually looks, having a Thorlabs Ganymede 2 high resolution OCT system that we've then added on lasers and modules to allow for both PAM and fluorescence microscopy imaging. We achieved sub-3 micron resolution for our photoacoustic microscopy, initially at half of the ANSI safety limit. And this is our initial characterization of that. We primarily do rabbit eyes. The reason being is that rabbit eyes have a similar size to human eyes. And since we're interested in clinical translation, we would like this similar size to assist with photoacoustic microscopy. And on D and E here, you can actually see the retinal blood vessels very nicely. You can see the branching pattern of the retinal blood vessels with high resolution. Now, one of the advantages I mentioned is you actually get deeper penetration than you can for strictly optical techniques. So here you see the deeper choroidal vasculature here. And this branching network is very complex. And prior to this, we really didn't have any appreciation or understanding of this complex network. And with OCT, you can see the 10 different layers of the retina labeled here. So OCT is very helpful for visualizing retina anatomy, and it's in standard clinical use nowadays. We then pioneered ultra low energy photoacoustic microscopy of the eye, taking it from 50% of the ANSI safety limit down to 1% of the ANSI safety limit. And we showed that you could get beautiful quality images with high sensitivity at even 1% of the ANSI safety limit. And this is one of our patents. We also can perform simultaneous imaging. So when we're doing multimodal imaging, one of the difficulties right now is most of our clinical systems are different machines. So you literally have to take a patient off one machine, put them on a separate machine, and it's very difficult to overlay that. So one of the advantages is not only can we do it all on the same platform, but actually we can do that at the same time to reduce artifacts and improve overlay for really truly multimodal imaging. We performed a comprehensive safety evaluation of photoacoustic microscopy, including electrophysiology, histology, immunohistochemistry, functional retinal imaging, showing no damage to retinal structure or function for out to a month after photoacoustic microscopy imaging. We can look at normal pigmented animals, see the retinal blood vessels with photoacoustic microscopy really nicely along with fluorescence. And we can develop these 3D maps where you can actually visualize with the changing slope of the back part of the eye, the actual course of the vasculature with really nice resolution. We can also perform several animal models. So this is a model of retinal vein occlusion where we actually inject in rose vangol. We occlude the blood vessels of the eye, and you can also develop retinal neovascularization resulting from it. And you can see the changes to the retinal vasculature very dramatically along with the development of new blood vessels. We can perform that in both kind of normal animals, can visualize the differences in these areas of retinal vein occlusion that you can see here in the treated areas versus untreated areas with significant changes in photoacoustic amplitude with that. And we can also see the development of new blood vessels, retinal neovascularization, and we can distinguish which ones are veins, which ones are arteries, which ones are the new blood vessels, and color code that to really get a full picture of the changes in the vasculature that are happening with these disease models. We can follow them longitudinally over time. So following it out to 3 months, we can see the dynamic changes in these blood vessels over time. In addition, we can see the development of new blood vessels or neovascularization in this model and follow it over time and see this really nice resolution imaging of these edges of the blood vessels going into the ischemic area of the retina where you have non-perfusion subsequently. This is another model. This is a model of diabetes inside the eye where we inject in VEGF and you get a very robust development of neovascularization. This is retinal neovascularization. And you can see here this fine network of retinal new blood vessels, completely different from the normal vasculature. And again, you can see the coursing of that over the course of the back part of the eye and see the neovascularization and follow that longitudinally over time. We can do that both in albino and pigmented animals, and we can actually quantify exactly where the blood vessels are, the size of the blood vessels, and quantify that the changes are in neovascularization rather than the normal blood vessels over time. We then have explored a number of nanoparticles, specifically gold nanoparticles, to enhance our photoacoustic signal. So these are unconjugated gold nanoparticles, not molecular imaging, but just contrast enhancement. Giving these intravenously and then looking at the imaging of the eye afterwards. And you can see that with gold nanoparticles, you can get significant enhancement over background blood in terms of the photoacoustic signal. You can use this to image the retinal blood vessels and get significant enhancement in the retinal blood vessels, along with the deeper choroidal blood vessels, and again, get significant enhancement in that. However, one of the downsides of conventional gold nanoparticles is the absorption peak corresponds to a similar absorption peak for hemoglobin. So we've also explored gold nanostars for molecular imaging. In this case, we have gold nanostars conjugated with RGD, a peptide that targets specifically new blood vessels and neovascularization. And here you can see choroidal neovascularization, and at 650 nanometers, you have very low endogenous signal. In fact, it's basically black here pre-injection. And then specifically, you can see where your new blood vessels are at the 650 nanometers. And here you see a 17-fold increase in photoacoustic signal. Now, one of the reasons why we love OCT for regular anatomic retinal imaging is one of the reasons why it's not so good for this molecular imaging is that there's so much kind of background signal with the different retinal layers that OCT signals increase by 167% only as opposed to photoacoustic. We then have explored some chain-like gold nanoparticles, which shift the absorption peak to 650 nanometers. One of the advantages of this is we can assemble or disassemble them as we would like. And here you can actually see really nicely that you can use these chain-like clusters conjugated to RGD to specifically target neovascularization and see exactly the extent of the neovascularization in these animal models of choroidal neovascularization here. And here you can see exactly in 3D the distribution of those nanoparticles and how it correlates with where the new blood vessels are. Whereas we use 578 nanometers PAM to see both where these new blood vessels are along with where our normal blood vessels are. OCT signal shows a lot of background noise, but is really nice for getting you the different layers of the retina. So multimodal imaging with OCT, PAM, and fluorescence provide complementary information. Photoacoustics very promising to non-invasively image blood distribution and can be achieved with high resolution at a safe laser intensity to visualize blood vessels in human-sized eyes. You can use it to look at retinal vascular pathology and then use nanoparticles to serve as contrast agents. And you can use this to follow longitudinally over 3 months, stem cells and precursor cells. And photoacoustic imaging really is the best way to do that. And this can enhance photoacoustic signal by 30-fold by following it over time. So I'd like to thank all the members of my group for their hard work. I'd also very much like to thank all of our collaborators in BME, specifically Xiding Wang, who I've been working with now for over 6 years. And it's really been a pleasure working with him and having those close collaborations, as I think other people have mentioned, between BME and retina surgeons and other clinicians like myself. I'd like to thank our sources of funding, and I'd like to thank you all very much for your attention, and I welcome any questions that you might have.
Thanks so much, Yannis. This was very cool. I couldn't help thinking during these first 3 talks at how each of these is really a nice example of the collaboration between engineers and clinicians and scientists, and how the questions are being driven by clinical and scientific needs. So quite wonderful. We have time for a question or two if anyone wants to unmute themselves or type in into the chat. In the meantime, let me quick ask a question. So the devices you use here, so how hard was it to get OCT and photoacoustics all together into one apparatus, and I presume, like, for example, the photoacoustic stimulation has to come in through the iris, and so it just seems like there's a lot to go in through one little hole.
Yeah, so certainly one of the advantages we have is that we can dilate the eye, so you can have a relatively large pupil. So typically, if we're working through a pupil, we have about a 6 to 7 millimeter pupil that we can work through, up to 8 millimeters or so. In some cases. So you do actually have some amount of space that you're working on. We started with an OCT system because there are a number of clinical systems approved for that. And then from there, we added on laser and fluorescence for that. And in terms of the technical difficulty of that, I would say it's definitely, it's readily possible. And I would say our hope down the line is to have more of a turnkey system. As I always joke around, I say clinicians are not as intelligent as engineers. We need something that's just a turnkey system that we turn on and it can take pretty pictures for us pretty readily. And that's what we're working towards. And I think that will be readily possible.
Right. So Jim Weiland, so is the acquisition time for the photoacoustics similar to OCT?
So you're going to be limited by whatever your light illumination source is. So right now we have a source that takes longer for the acquisition of the photoacoustic microscopy than the OCT, but that's really limited by our laser light source. So I would anticipate in a clinical system down the line having a much faster acquisition for photoacoustic microscopy that could just take a couple of seconds. But right now, to get a large volume cube like what I was showing, typically would take about a minute or so for our photoacoustic microscopy setup. But down the line, our anticipation in our newer system that we've actually developed is on the order of a couple of seconds. Photoacoustic microscopy is not necessarily inherently any slower than OCT, but in the current iterations of it, it tends to be a little bit slower on the order of seconds rather than sub-second like OCT typically is, Jim.
Very good. Well, thanks so much. I think in the interest of time, we'll move on to the next part of this session. As you guys have probably seen, coming up next, we have what we might refer to as power pitches or something of that form, where basically they're short little pitches on dissertation topics from our students. We have 8 of them lined up. This will be done pretty efficiently because they've all been pre-recorded, and Brandon is going to be playing those for us. I think many of the students are online here, so feel free during these presentations, if you have a couple questions, feel free to type them into the chat window, and hopefully if the students are here, they can reply through the chat. Okay, so Brandon, you all set? Okay.
I'm Eric Hobson, and I'm advised by Dr. Sherry Dang and Dr. Jan Stegman here in BME. Today, I'd like to talk about a technique for the non-contact characterization of viscoelastic biomaterials that we call resonant acoustic rheometry, or RAR. This technique is non-contact, utilizing ultrasonic pulses to both generate and track resonant surface waves on soft biomaterials containing the cylindrical wells of a microplate. This approach is very high throughput, with each measurement taking less than 1 second. In the lower left corner, you can see an example of these acoustically generated surface waves that we captured using a high-speed camera. These oscillations are tracked using echoes from repeated ultrasonic pulses, and we use spectral analysis in combination with hydrodynamic models of surface waves to calculate material properties such as the surface tension, shear modulus, and viscosity. Validation of RAR measurements was performed using shear rheometry, a more conventional approach, for a variety of tissue engineering materials, including fibrin, gelatin, and agarose, and we saw excellent correlation between these two techniques. In addition to static measurements, RAR can be applied to the measurement of dynamic material transitions. These rapid, non-contact measurements can be applied repeatedly to samples at almost any time interval, and can even be used to track multiple samples simultaneously. We've been able to use this to track rapid dynamic changes such as the UV cross-linking of PEG-norbornene, where multiple samples were taken per second to look at this quick liquid to solid transition. In addition, we've been able to look at much slower processes, such as the genopin cross-linking of gelatin, where we took longitudinal measurements to look at mechanical changes that occurred not only over seconds and minutes, but over hours and even days. In summary, RAR can be used to characterize the mechanics of soft biomaterials in both static and dynamic states, and has a number of advantages over conventional approaches, such as its non-contact and high throughput methodology, and the fact that we can still generate these quantitative viscoelastic measurements. Thank you.
Hi everyone, my name is Kai-Wei Chang. My advisors are Dr. Xiaotian Wang and Dr. Guan Xu, and my topic is photoacoustic tomography of visually evoked cortical responses in the mouse brain. So what is photoacoustic? Photoacoustic phenomenon is a combination of optical excitation and ultrasonic detection. When a pulsed laser is illuminated on the tissue will absorb the laser energy and cause thermal expansion. And this thermal expansion will generate ultrasonic waves to be detected by ultrasonic transducers nearby. Then image reconstruction can be done. Photoacoustic technique is sensitive to endogenous optical absorption contrast, such as red blood cell, lipid, and collagen, which are selected by wavelengths. Therefore, can achieve functional brain imaging according to neural hemodynamic changes. Also, due to low scattering of acoustic waves, it has a higher spatial resolution even in deep tissue than other pure optical imaging technique. So in this work, we apply photoacoustic tomography to image the whole mouse brain using a 256-element fluorine array and use white light to stimulate the retina of the mouse and monitor its cortical response in the primary visual cortex. The top right figure is the reconstructed photoacoustic image. The vasculature perfectly matches the photograph. Then we extracted the relative change in the primary visual cortex area and compare two different types of mice, the wild-type mice and the retinal degenerate RD1 mice. During the light stimulation, the cortical response can be clearly observed. The peak intensity and peak delay were calculated for statistical analysis, and significant differences were found between the wild-type mice and RD1 mice in both peak intensity and peak delay. The results prove that the photoacoustic tomography is a promising imaging technique for brain activities and can be potentially used to study brain disease models. So this is my presentation. Thank you for listening.
Hello, everyone. My name is Xiaotian Tan. I'm currently a postdoctoral research fellow from Professor Xu Dongfan's group. Today, I'm going to introduce our recent work on the rapid quantification of COVID-19 IgG for convalescent serum evaluation. A few years ago, our group has developed this fully automated microfluidic chemiluminescent ELISA system. And with our system, we were able to use only 8 microliter of sample and reagent volume to perform an ELISA, which is more than 10 times smaller than the required sample volume in a traditional plate-based ELISA. Also, thanks to the high surface to volume ratio, we were able to speed up the immunoassay by around 10 times, also comparing with the traditional plate-based ELISA. After the start of the COVID-19 pandemic in 2020, we initiated this study, which is to quantify the abundance of SARS-CoV-2 specific IgG in recovered patient samples. The first thing we did is we evaluated a panel of 4 antibodies that were reported to have high binding affinity towards the spike protein of SARS-CoV-2. What we found out is D006 is in fact excellent antibody, an excellent calibrator antibody for SARS-CoV-2 spike protein because it has the widest linear dynamic range towards this protein. And with this calibrator, we were able to detect down to 2 nanogram per ml level within only 15 minutes of total assay time. We also applied this approach to the actual patient samples. What we found is the antibody abundance in the patients who reacted strongly towards the virus is about 200 to 300 times higher than the patients who reacted only weakly toward the virus. And the vaccine recipients, such as myself, is having a very similar antibody abundance toward comparing with the patients that reacted strongly toward the virus. This may be able to serve as an indication of the vaccine's protection efficacy. Thank you very much for listening.
Michelle, and I'm a PhD candidate in the functional MRI lab. Today I'll present our recent work on connectome-based predictive modeling of cognitive measures in mild cognitive impairment. Functional connectivity can be measured as correlated brain activations from the fMRI bold signals of different regions of the brain. Recently, much work on the analysis of FC aims to use it as a type of fingerprint, unique to individuals and reflective of physiological changes in the brain, to form models that predict related behavioral measures. We have evidence that FC is interconnected with behavioral changes, such as those that occur with neurodegeneration, making connectome-based models a good candidate for prediction of cognitive changes. 2 studies have compared different types of task and resting state data and different types of models looking at a given condition and its resultant deficits. So this is an area of ongoing investigation with significant clinical relevance for neurological and psychiatric diseases. We aim to compare different modeling approaches, including a new adaptive version of PLS regression in their abilities to predict cognitive measures in subjects with MCI. Since PLS regression uses both predictors and responses to estimate the model, it is an attractive approach for representing the interplay between behavioral deficits and functional connectivity in a cohort on the continuum of MCI. Shown here are results from PLS regression using feature selection called PLS beta. This approach identified a relationship with object location task-based fMRI data and the R-bands delayed memory index that dropped out of significance with permutation testing at 1,000 iterations, and also identified high-degree nodes in the frontal lobe, a region thought to be involved in compensatory memory for those with MCI. Further testing will expand to different types of fMRI task and rest data and other cognitive measures. Thank you for listening.
Hello everyone, my name is Christopher Tossas and I'll be presenting on computational hemodynamic modeling of pediatric cardiovascular diseases. These diseases are a significant burden to our healthcare system, affecting 1 out of 100 individuals born in the US and often require lifelong medication and surgical interventions. And computational modeling can be used to help with diagnosis and treatment of these diseases. And in this talk, I will be focusing on renovascular hypertension and an endovascular repair of a Fontan surgery in a patient with hypoplastic left heart syndrome. These models are informed by clinical data on anatomy, flow, and pressure, and can be used to provide insight on quantities that are not easily accessible through this data. And these models are used to perform disease research as well as aid in surgical planning. To study renovascular hypertension, models were constructed using data from a patient with an abdominal aortic stenosis, shown here in areas of higher velocities, as well as a significant pressure drop. And we discovered a presence of high-frequency disturbances in renal artery hemodynamics, and therefore we hypothesized that these disturbances contribute to the persistence of hypertension after these patients have undergone a successful surgical repair. To aid in surgical planning, we collaborated with the pediatric cardiology team at BOTS, who is considering an endovascular repair on a patient with hypoplastic left heart syndrome and a dilated Fontan pathway shown here. We simulated the endovascular repair with realistic material properties and saw that this repair led to increases in venous and pulmonary arterial pressures, as well as an increase in venous reflux to the liver. And these changes were deemed unfavorable to the patient, and therefore the clinical team decided against performing the endovascular repair. Thank you so much for your attention.
My name is Sarah, and I'm a second-year PhD student in the Jew Lab. One of the major research projects in our lab is investigating histotripsy as a method of non-invasive brain treatment. Histotripsy is an emerging technique that uses short, high-intensity ultrasound pulses to generate inertial cavitation in soft tissue. When the peak negative pressure generated by an ultrasound pulse exceeds the intrinsic medium threshold, a bubble cloud in the tissue expands and collapses, inducing lethal strain on the cellular components of the surrounding tissue. Histotripsy has shown promising results in the field of transcranial ultrasound therapy. Our group previously demonstrated that a 500 kHz histotripsy array, shown on the left, is capable of precisely ablating brain tissue through the skull with single-point ablations, a grid of points, or contiguous lesions without significant heating of brain or skull tissue. To this end, our group is currently investigating the effect of histotripsy on brain diseases such as intracranial hemorrhage and glioblastoma multiforme. To investigate the effect of histotripsy on immune response in glioblastoma, we injected immunocompetent mice with tumor cells, monitored tumor growth via MRI, then treated some with histotripsy and left the rest as non-treated controls. Tumors were treated using a 1 MHz ultrasound transducer with 1 Hz pulse repetition frequency at a peak negative pressure of 32 megapascals. 2 days and 7 days after treatment, we harvested the treated and untreated brains for histological assessment and flow cytometry. As shown in the histology image on the left, it was found that histotripsy successfully ablated part of the tumor by homogenizing brain tissue and killing intact tumor cells. Flow cytometry revealed that 6 types of immune cells were either significantly increased or were trending upward after histotripsy treatment. Our results show that histotripsy is a promising non-invasive method of stimulating an immune response in glioblastoma mice, which may lead to survival benefit. Thank you for listening.
Hello, my name is Kritika, and I will be presenting a data-driven method for coronary reconstruction. The motivation for my project is to develop an image-based computational method to determine FFR, which is defined on the slide here as the ratio of pressures on either side of a stenosis and is a clinical metric used to assess coronary artery disease severity. To do this, I have a pipeline that starts with X-ray angiography images, followed by a neural network for automatic vessel segmentation, a 3D vessel reconstruction algorithm, and finally, a 3D computational fluid dynamics simulation using our lab software Crimson to compute FFR. I'll be focusing on the first two steps today. For the first step, I've developed a neural network called AngioNet, which is composed of an angiographic processing network, which you can see here, that works in tandem with DeepLab v3+, a state-of-the-art neural network for vessel segmentation. So how does AngioNet improve DeepLab v3+? We can see several challenging examples here, where AngioNet is able to identify more of the vessels than DeepLab alone, and is also able to successfully ignore the catheter. If we look at the table below, we see that AngioNet's accuracy or mean dice score is significantly higher than DeepLab alone. For the second step, I have my 3D reconstruction algorithm, which is a semi-automatic back projection algorithm. For this, we start with 3D printed models of the coronary tree, as these are known geometry. We then take X-ray angiograms of these and feed them through AngioNet to produce binary segmentation images, where a user will select which branches correspond to each other in both images. We can then map these image planes into 3D space, as seen in the top right. And finally, we can ray trace these X-rays to identify their 3D points of intersection and compute a center line seen in the bottom right. Thank you.
Okay. Well, thank you to our students. We're only about 5 minutes behind schedule, which is that in itself amazing feat. So thanks, Brandon, for having these pre-recorded. That was a lot of fun. I'm always a sucker for student presentations, and it was fun to see the diversity of topics here. So in the last part of the imaging session today, we're going to have a panel discussion. We have an illustrious panel with we have Vikas Gulani, Sherry Dang, Zhen Xu, Xuding Wang, and Jeff Fessler are our panelists for today. And so I know Zoom meeting here works in unusual ways sometimes. And so one suggestion I might have, if you want to see our panelists, is that you can, all three dots in the pictures there, you can hide non-video participants, and at least you'll get the all of our participants of this will actually be front and center on the screen. You'll be able to see each other, and whoever else wishes to be seen by everyone else, like Christopher. So with that, I think we can get rolling with this session. I think I'd start out by inviting our participants each to introduce themselves briefly and tell us, very briefly, because we want to get on with the session, but at least tell us what your research interests are, where you are, what department you're with, and so on and so forth. And we'll start with Dr. Gulani, if you could.
Hi, I'm Vikas Gulani. I'm the chair of radiology here at University of Michigan. I apologize for the bright light coming from behind me. I'm trying to adjust for it, but having a hard time. But I am an MR researcher by core interest, and I've known many members of BME here for a long time. So it's my pleasure to join you today.
Great. Thanks, Vikas. Zhen, do you want to go next?
I am Zhen Xu, professor of biomedical engineering here, and my primary research focus is ultrasound therapy, and particularly we're developing histotripsy for cancer, neurological, and cardiovascular applications.
And we're following the order on my screen. I apologize. So Xuding, you're next.
Hi, I'm Xuding Wang. I'm a faculty member in BME and also holding an adjunct faculty position in radiology. My research has been focused on photoacoustic imaging, and I try to also go a little beyond that. So recently, we're also doing a lot of research on ultrasound therapy enhanced by laser.
Great. And Jeff?
Hi, I'm Jeff Fessler. My primary appointment's in electrical engineering and computer science. but I have secondary appointments in BME and radiology. In fact, I've been working with BME since even before it was a department, many, many years ago. And my primary research interests involve image reconstruction problems in PET, SPECT, CT, and MRI.
Right, and Sherry?
Hi, my name is Sherry Dunn. I'm a professor in the BME department, and my research interest is in the general area of ultrasound. I'm more interested in biophysics and ultrasound techniques that involve, I guess, cellular biophysics and stuff like that, and biomaterials.
Fabulous. So I hope we can get a robust discussion here. I know it's hard when you can't pass the microphone back and forth in literal sense that we're used to doing it, but hopefully we can chime in as and if for our panelists, if you need to if you want to speak up, just raise your hand or give me some other signal, and we'll try to make sure you get your say in. So I actually thought I would start out by posing a question to the panel, which is, what do you think? What are some of the grand challenges in imaging over the next 5 to 10 years? So if I had a new student coming in, what should I have them be looking at? And so and Vikas, I'm going to start with you if you don't mind, because just have you kick it off, because as a department, you're supposed to be thinking strategically all the time, and so.
Well, thanks for that intro, but I don't know that I have any extra wisdom, but I will say that my view is that we have been a bit of a victim of our own success in some ways. The CT, ultrasound, MR, PET, these are all ubiquitous now. So the barrier of entry of a technology finding its way to translation, this is getting higher. And that said, the merging of structure and function, it remains quite nascent. The only thing we measure in clinical practice right now still is distance, basically, the size of things. And there's a few things that we measure, but they're very unusual. So there's a lot of opportunity to be quantitative and to be merging both anatomy with something else, or some sort of function. Also, targeted drug delivery and treatment remain super needed and are still nascent. And we still tend to do a lot more damage than we want to when we go around treating patients. And so I think that there's a big area here. A third thing that I would encourage us to think about is that we've had a rather privileged view of technology in imaging. That is that we have been designing for the rich primarily. And so we don't really seem to worry about the cost of a technology, because we say, well, it's new and it will mean something. However, most of the world doesn't get access to our technology as it is. And if we're designing primarily for the rich, most of the world will remain distant from our technology. I think the opportunity exists to think more globally. And if you start thinking about how to reach populations that perhaps can't afford the technologies we have, it will force a thinking in how we design technology in the first place, and become an impetus for innovation. Those are some thoughts that I think that engineers ought to be thinking about from the perspective of how we use imaging.
Any of our other panelists? Great, thanks, Vikas. Want to chime in, or I'm going to call on you if you don't. So,
I will prevent Doug from calling me. I'll volunteer. So I think the imaging traditionally people are thinking has been used for diagnosis, which it has been doing a great job. And traditionally our engineers have been thinking about when you do imaging is to make it better resolution, better quality, more quantification, and lower cost, which I still think continue going that way is important. If anybody can make MRI as as low cost as ultrasound, that would be great. But on the other hand, I think there's a whole trend of new areas that are coming up, and towards not only by medicine to go towards non-invasive, and also other applications. So there are image guided therapy, right? So imaging is not only just used for diagnosis, and also for guiding certain treatment that's non-invasive. In my lab, we're developing histotripsy or ultrasound therapy, but there are more there are laser therapy. There are different forms of ablation therapies. Even radiation therapy, they are looking for more some imaging feedback, and so imaging guidance and monitoring for the newly developed minimal invasive or non-invasive imaging therapy also are a kind of new area. And people are talking about immunotherapy and all this. So you have all this new treatment now also require follow-up, right? Because you don't want to always, you can cut the patient open. Biopsy, there's some limitation. So imaging still has its advantage. Then going beyond medicine, there are also tissue engineering, some biology that require imaging too. I shouldn't say require, but there are new applications out there. So I think the more I think is not only challenges and also opportunities for imagers, that there are a lot of applications out there and new ways of doing things. Now we cannot just sit and only thinking about making imaging better. We have to think about how do we utilize the technology to kind of adapt to this new world of emerging applications. So that's one thing. And the second, I think the grand challenge, another thing is big data, AI, right? All those incorporating into things. So now that when you get imaging, you can get tons of data now. And the people are talking about using the artificial intelligence. How do you deal with those large set of data and getting useful information out of those? So I'll stop there.
Great. Go ahead, Jeff. I saw you threatening.
Well, Xiaoting unmuted before.
Okay.
So go ahead, Xiaoting.
Oh, okay. Yeah, so I agree with everything said by Jen, and because so imaging is really a tool to help other areas in medicine, right? So I think in the future, the main value of imaging is still reflected by helping in diagnosis and helping in treatment. So that's no doubt. But to me, of course, there are a lot of hot topics in medical imaging, but to me, I'm super excited about two topics. The first one is the single cell and single molecule level of imaging. So for example, if you can track the cells in vivo in human body, CTC cells, and try to understand their interactions with the immune system in vivo, non-invasively, that's super exciting, right? Also, like stem cell therapy is a super hot topic right now, but for most of the stem cell therapy, after injecting the stem cells, we don't know what they're doing. And if you can have some imaging technology which can track the stem cells to understand their PK/PD, that will be really a groundbreaking technology. And another topic I'm super interested in is the brain mapping. I mean, brain initiative is still running and a super hot topic. As you can see, several talks today are all focused on brain, and this is still a wonderful area for imaging researchers to get in. So I will stop here.
Okay, I just want to say a word about computational methods as a way of looking forward. Partly by starting by looking back. When I came to Michigan in 1990 as a postdoc, some people were negative about the use of iterative reconstruction for PET and SPECT because it was so compute intensive. And then by 1997, the companies all had methods for iterative reconstruction in PET scanners. It took us 15 years between the key publication and when it finally reached FDA approval and widespread clinical use. But now every PET and SPECT scanner today uses iterative reconstruction. Pretty complicated methods, really. I mean, computers got faster and people's understanding of how those methods improved or affected the image quality advanced over time. Similar story with CT. In the early days, everybody said, oh, those methods are too slow. And then by about 2012, companies were selling it. And then even faster trend in MRI, like the key compressed sensing paper was published in about 2007, and by 2017 we had FDA approved compressed sensing methods that are available commercially today. The trend with deep learning was even faster, right? It was 2017 or so when that became the buzz, and then just a couple of years later already companies were commercializing, getting FDA approval for deep learning kind of methods. And so my advice to students would be, no matter what modality and what you're thinking about, I would not focus on sort of cutting corners to make an algorithm slightly faster if it compromises image quality, because a year or two later, that algorithm will be obsolete. Focus on trying to get the best image quality you can and trust that eventually the computing methods will get fast enough to make your sophisticated algorithm practical, because we've seen that over and over across modalities. Cool. Of course, somebody has to say something about deep learning because it's such a hot topic that has been used in all modalities. It's very exciting, but I think there's very significant challenges. I'll just refer to some joint work that Doug and I have done with one of our graduate students, very exciting work on optimizing the trajectories and the reconstruction algorithm for MRI. Interestingly, in that work, we show that you get, this Guanhua shows that you get different optimized sampling patterns for the knee and the brain, which is exciting and interesting. But if you think about, if Akash were to think about how many different protocols are on an MRI scanner, hundreds, if not thousands of protocols, if we have to separately train and optimize different trajectories for every single protocol, that's a real significant undertaking. And so developing methods that are really generalizable across different scanners, different modalities, I think is a huge challenge ahead for our field in the deployment of deep learning methods, especially maybe on the image formation side, but probably on the image processing side as well.
Very cool. And Sherry, did you have something you want to say?
Do I have to? Well, I think everybody said everything that's important. So I'm just going to add one part that is, I don't know, it's something that I've been thinking about. I think, for example, in the case of ultrasound imaging, and there are like lots of research in the field, and doing all kinds of things on clinical systems. But I think one of the promising, very interesting area to do is to make, I guess, to adopt the machine learning, AI and stuff, but in a very small system. So like a handheld devices. And so the handheld devices, there's a very recent one that a company went public, probably just a month ago or so, called Butterfly. So it's just this one handheld system, just the size of your iPhone, and then can do, the images, generate pretty good images with, the image processing algorithms that have been developed. So that's, I think it's a very interesting area for people, students who are interested in ultrasound imaging, not just the ultrasound acoustic engineer part, but it's a combination of computer science and data, data processing and data science and AI and stuff all combined together and put this system together. So a very good quality and high-speed imaging in a very small system. So the Butterfly system actually was taken to a space station a month ago. So you cannot take, an ultrasound system up to the space. So I think it's a very interesting area or direction, because it combines not just the acoustic engineers, but all the other, hot areas, AI, deep learning, and computer science.
Yeah, I agree. I mean, I think accessibility is one of our grand challenges for sure. Something that's been creeping around in the MRI field is the so-called 5-minute exam, and I actually think that's in MRI worlds, a 5-minute exam is sort of shocking, but it actually does work and has clinical utility, and I think that's pushing that and making, using that as a way to increase accessibility to the technology, I think is one of the things that at least in the MRI side would be extremely valuable. If I could actually direct the question back to Vikas, you talked about function and anatomy, and I would like to, there's been some push for MR PET scanners and and PET CT scanners, and I'm just wondering what's your perspective on those as a way of combining function and anatomy, and if there's other opportunities like that are exploitation.
Yeah, I mean, PET CT is now pretty ubiquitous. The FDG PET CT is It's built into our clinical routine, and you can't get rid of it. And there are multiple areas where we're headed in PET-MR, but PET-MR has sort of lagged a little bit in terms of its killer application. What can you do with it that you can't do with a PET-CT? I think this is developing because dual use tracers and things like that will probably make a dent there. I think that if PET-MR, for example, ends up being a really expensive PET-CT, it won't take off. It has to have something new, something different that it provides. But I think in general, both approaches and these multimodal approaches are trying to get at providing the best of multiple worlds, that you have some spatial information at a high resolution that you're interested in, and then you have functional information that has to be mapped very exactly to that spatial information. And I think that's something that has been lacking, and we're only starting to make a dent there. And I think this is going to take off a lot more. I think PET-MR is kind of a baby right now, but in 10 years, I'm really curious as to where it ends up, how much better we can do into the future.
By the way, I would like to sort of, feel free for people to type in questions as well, and I can relay them, or we're actually going to probably go on for maybe about 5 more minutes. We're running a little bit over, but so feel free to pipe up if you would like to have our audience respond to things. I think another thing that was mentioned multiple times here was involvement of imaging with therapy, right? And that I agree that appears to be a trend overall. I know there are now MRI scanners in surgical suites, although it's been reported to me that those end up being more surgical suites than the imaging part is not actually always even used. And so it seems to me that there are lots of opportunities along multiple modalities. I was just wondering if any, besides monitoring therapy, but perhaps directly in the therapeutic loop itself, does anyone want to a little bit farther?
So since I kind of presented the idea, I follow. I don't want to specifically comment on like going too deep into therapy, but I have always thought about this idea. We have a bunch of imagers here, so I think probably a good opportunity to ask this. I have been working at University of Michigan for 20 years. I still do not know the full extent of all the capabilities we have on campus. And we have been talking about how to facilitate collaboration between engineers, imagers, clinical side and the application side. I'm wondering that if we can put a central list together of the imaging capability that's available for research at least. So people who want to use it and think about application, they want to use it, not only just the instrumentation, but also the experts with expertise in those areas. Really just a website, a central list. So people who are thinking of doing therapy and want to look for collaborators to look at the imaging for monitoring, for example, they can go to one place, then take a look and say, okay, I can contact this person who has this capability or this instrument equipment. Right now it's still about you have to know the know-how. So you have to be on campus for a while and know where to go. We have people here. I think it's possible and it wouldn't take a lot of effort to just have a simple web page. Sorry, it's not a direct answer to Doug, your proposed question, but I think it's a good opportunity to bring up.
Good discussion. Yeah.
Yeah.
I concur with Jen. I remember before the COVID pandemic, we have some imaging theme meetings, right? And this is a topic that we discussed during the theme meetings. I think it's a good idea to have a centralized imaging core or imaging platform so that everybody understands who are doing what. And to be honest, not just the radiology, not just the BME, there are also many other researchers in campus doing imaging. For example, in ophthalmology, there are a lot of people, right, doing imaging, internal medicine, for example. But Yeah, I agree with Shen. I have been working in Michigan also almost 20 years, but I'm not sure I know everyone doing, and I don't know what are in everybody's lab. So I like that idea of like a centralized website and also like periodic kind of symposiums. Actually, I talked with Brian Folks several times about like a kind of a grand round, imaging grand round, so that during every month or every 2 months we can get together, right, more frequently than this BME symposium, and people can exchange ideas.
I mean, certainly for me, coming back to Michigan, this was one of my central goals here. And an attraction to come to Michigan is the fact that the medical center, the engineering campus, letters and sciences, everybody is very close to one another, actually physically, compared to most other universities. And this is a massive strength. Very few places have it. I can think of Stanford and UCLA. There's not a lot more. And there's it's rare. And we're not making enough use of it, in my opinion. And this is something that's been a central goal. Certainly one of the reasons we formed MIT, which left out MRI from the name for a reason. And that is to try to pull together many interests on campus together. There's also, I mean, to complete the list, there's public health, there's business, there's multiple people looking at this from angles. So I can certainly volunteer the services of radiology to try to pull such a list together. I am very supportive of such an effort.
Yeah, I think here, at least, because if you can put together the radiology resources, we can put together BME resources, then even those two together would be a pretty long, big list and a great starting point. And I think also when you have those kind of web page, then we're not limited to just faculty, right? Like even the students and the trainees can go on. And I have been always amazed by the collaboration formed by actually the trainees and the students. That they go and they talk to each other. I think it will help us expand the scope quite a bit. And I totally agree with you, because that we really have a very strong imaging related program here, and we have not been able to fully utilize all the potential, and we should do something about it.
Yeah.
Sorry, Doug, we use your 5 minutes and you're muted.
That's fine. I think at this point we're a little bit over time, but not terribly. So I'd like to just give an opportunity for last comments that anyone has or questions from the audience. So here's sort of a fun question. So with AI coming, how long is it before we're going to replace all of us, including the radiologists? From
Never.
Considering that 90% of the world hasn't gotten access to most of our technology, I don't think we're in that danger at the moment. I think we'll replace the ones that become obsolete if we allow ourselves to become obsolete. But I think if we keep innovating, I don't see us being replaced.
That's a great comment to finish on. So I want to thank the speakers for the contributions. I want to thank the students, thank our panelists for participating. I'd also sort of like to remind everyone at 4 p.m. today, there's a poster session. We have some posters in imaging, as well as some posters in neuroengineering. And what time is the next session? The
2 o'clock.
Karen?
Neuroengineering, 2 o'clock.
2 o'clock. So hopefully you've registered for all of the sessions, and that should be a great session as well. So thank you, everyone. I guess we'll call that a wrap.
Thank you. Bye.
Thanks, everyone.